All right, so we've talked so far in this chapter about different types of learning that occurs, different types of memories, but how is it that our brain actually learns new things and remembers them? At the neural level, memory is associated with changes that take place at the synapse. So again, remember, the synapse is where one neuron influences another neuron. So to find neural correlates of memory, we then have to really look at this very basic synaptic level change. So we can determine how synaptic changes are correlated with memory in the mammalian brain. We can also localize the synaptic changes to specific neural pathways and analyze the nature of synaptic changes themselves. So how is it that we would do this? One way is long-term potentiation. So long-term potentiation in response to a stimulation at a synapse changes the amplitude of an excitatory postsynaptic potential that lasts for hours to days or longer. So you might want to think back, because we talked about some of this stuff earlier on when we were talking about EPSPs and IPSPs and sensitization and desensitization. So in response to stimulation at a synapse, you can have a changed amplitude of excitatory postsynaptic potentials, so they can increase, and this change in amplitude can actually last for a while. So this plays a very important part in associative learning, linking of two or more unrelated stimuli to elicit a behavioral response. So when your brain is starting to learn, and if you've formed an association that might be relevant, then one neuron is going to try to have a greater and increased effect on another neuron. So if that first neuron can cause that second neuron to fire, then it can result in long-term potentiation. And this basically, what would happen then is that the firing of the first neuron connecting with the second neuron would result in an increase in excitatory postsynaptic potential, and this then increases the likelihood of the second neuron passing on that information to a third and fourth neuron, and so on. So if you have a stronger relationship, basically if they fire together, then they wire together. And so that long-term potentiation is sort of that increase of like, okay, we fired together. And now if that first neuron gets stimulated again, then it's going to have a larger effect on that second neuron, which is going to be more likely to cause that message to be passed down. And then if that first neuron gets activated again, it's going to have even larger amplitude of excitatory postsynaptic potential. So this increase in postsynaptic potential, this is the sort of longer term potentiation, and it can last for quite a while. Here's an example. So if you have a strong burst of electrical stimulation applied to the presynaptic neuron, this produces an increase in the amplitude of the EPSPs in the postsynaptic neuron. So this is basically evidence that one neuron communicated with another neuron. It's kind of the neuronal equivalent of a paper trail. So each dot here represents the amplitude of the EPSP in response to 1 week test stimulation. Okay, so 1 week sort of stimulation, one small sort of like, okay, fire a little bit here, fire a little bit there. So they're all just sort of like weak little tests to try to stimulate it here. If you have this period then of stimulation where you're going to stimulate the nerve even more, then you have this increase in the amplitude of the EPSPs. And if you notice here, this increase persists, okay, well beyond time. So it's all down here, and it all sort of moves up, which basically means that you have a stronger paper trail, or this first presynaptic neuron is more likely to make this postsynaptic neuron fire. For EPSPs to increase in size, more neurotransmitter must be released from the presynaptic membrane. The postsynaptic membrane must become more sensitive to the same neurotransmitter, or both changes must take place. So again, we're returning back to some of the content that you learned earlier in the course. you know that EPSPs, if you have enough of an EPSP, that causes an action potential. So if an EPSP is going to increase in size, that is going to increase the chances of the postsynaptic neuron firing. So here we would have a sort of weak electrical stimulation. Because the MDA receptor pore is blocked by magnesium ion, release of glutamate by a weak electrical stimulation activates only the AMPA receptor. So we have just a little bit here, okay? So we have this glutamate kind of comes out here, and you have just the MDA receptor. This is your presynaptic, and this is your postsynaptic. Now, if you have this stronger electrical stimulation, okay, what this does then here is a stronger electrical stimulation can depolarize the postsynaptic membrane sufficiently that the magnesium ion is removed from the MDA receptor, okay? So you can see here it's sort of blocking it. If it's strong enough, then it's removed. Then what this happens is that you have more information that can come in. So this neurotransmitter can act as a second messenger to cause the formation of additional receptors. So again, you have weak stimulation, which doesn't really do a whole lot and sort of functions as normal. And the neurotransmitter leaves, it activates the receptor here. But if you have a very, very strong stimulation, this results in a long-term potentiation. You have more neurotransmitter that leaves. This increase in neurotransmitter depolarizes the postsynaptic membrane sufficiently that this magnesium ion is removed from this receptor pore. So then it opens up this receptor pore. The neurotransmitters can come in, activate this second messenger. And if you recall, second messengers, they can instruct the formation of neuroreceptors. So after this really strong electrical stimulation, if you have just a weak one, the glutamate now released by this weak stimulation can activate the NMDA receptor to allow the calcium influx, which through a second messenger increases the function or number of the AMPA receptors or both. So weak doesn't do a whole lot, but then after something very, very strong, then that can cause changes which can allow even weaker stimulation to result in firing of the neuron and the passing of this information downstream. Long-term depression, or LTD, decreases EPSP size. So if LTP is a mechanism for creating memories, perhaps LTD is a mechanism for clearing out old memories. A neuron becomes less active in response to repeated stimulation. So this can also be something that could, again, occur with some habituation, or if you're trying to have extinction. So you've learned something, but now you're learning that at one time this stimulus resulted in more and higher EPSPs and resulted in stronger association between these neurons. But now the stimulus is no longer predicting this positive outcome, and so then you could have this long-term depression, which would actually result in a decrease in the EPSP, sort of this less predictability, and you can try to have extinction so that you can form new, more adaptive associations than the ones you currently have. So if you wanted to modify your existing memory circuits, then neurons basically need to change their structure in response to their changing experiences. You can have changes in the number of dendrites. This can be used to infer synaptic changes. More dendrites equal more connections. You can also have new synapses between neurons that are already connected or between neurons that were not previously connected. So again, neurons need to change their structure so they can have changes in dendrites, and they can also have the formation of newer synapses. So let's learn a little bit about that here.
You would not be here if you weren't interested in learning, and neither would I. But here's something we haven't learned about together: Learning, the ways in which we acquire and retain knowledge, which is the very definition of learning, is really a science in itself, and like any other discipline that involves the study of the human brain, is practically still in its infancy. Just 20 years ago, most scientists believed that once we reached adulthood, our brains were pretty much fixed. Not that we were incapable of learning anything new exactly, but the assumption was that our brain's development phase was over, and now it's pretty much there to remind our hearts to keep beating and occasionally let us remember where we left our phone. But thanks to huge advances in things like functional brain imaging, we have a clearer picture than ever of how our brains work, and we're beginning to observe some wonderful things. For one, we now know that the process of learning actually alters the structure of our brains at the cellular level. And what's more, it turns out that our brains never stop changing to make room for new information. People often compare the human brain to a computer, but imagine a computer that can actually grow new circuits as it acquires new facts and associations, and you have a much more awesome comparison. This is the gift known as neural plasticity. You might think of plastic as being stiff and cheap, but in biology, plasticity refers to the capacity of living things to mold themselves to new conditions, and our brains are great at it. For one thing, your brain cells, or neurons, are always changing their connections to one another to meet changing demands. Each of your neurons consists of a central body with spindly dendrites and a long axon stemming from it. The neuron transmits electrochemical signals to other brain cells through its axon and receives signals through its dendrites via connections where the cells meet, called synapses. When you're born, each neuron in your brain has about 2,500 synapses connecting it to other cells. But by the time you're 3, and you've become just a sponge of fascinating new information, like what sound a piggy makes and where your mom's face goes when she hides it behind her hands, you have 6 times as many, up to 15,000 synapses for each neuron in your brain. In this regard, it's kind of true that your brain hits its peak when you're young, because by the time you're an adult, your brain cells have about half as many synapses as when you were 3. But it turns out that's okay, because we now know that synapses just shrink up when they're no longer needed. Like, you know now that pigs go oink and that people still exist when you can't see their faces, so you don't need to keep those connections to remind you. This process of winnowing down unused connections is known as synaptic pruning. Meanwhile, your neurons experience all kinds of new growth as you continue to soak up new information, like how to drive, how to solve for X, and how to get through that one really hard level in your favorite castle smashing game. Here, the key to learning is memory, because you need to retain that information in order to apply it in the future. So your brain cells can change in different ways depending on how long you remember what you've learned. As you stockpile data in your short-term memory, for example, the structure of your existing synapses has been found to change, with more and stronger dendrites growing to reinforce them. So for info that you retain for just a short time, like how to destroy digital castles using rocks and fire, you don't need to sprout whole new connections. Just beefing up the existing ones you've used so far will help you master the game just fine. But when it comes to important stuff like the learning we do at school, or hopefully the stuff you learn here on SciShow, your neurons actually forge entirely new synapses over time as you relearn, re-remember, and reuse the information. This is how your brain builds the long-term memory you need to retain the learning you're doing right now at work and at school, and turn it into a lifetime of applied knowledge. So I said it before, and I'll say it again. Take care of your brain, and odds are, it'll take care of you. Thanks for watching this SciShow Dose.
So I thought that was a fun little recap of sort of putting all of the pieces that you have learned together in terms of your changing brain, the role of dendrites and synapses and neurons and learning and maintaining information. Now to get a little bit more specific, okay, the video talked about dendritic plasticity. So if you were to form new connections between neurons, then this can happen at the synapse level or at the dendrite level. If you look at how this neuron changes, if it's labeled with dye, and you look how this neuron changes 88 to 90 days later, you can see that an actual cell body is relatively same, but here you have this greater branching of the dendritic spines. You see similar here that you have a fair amount of branching, and again, days later, this branching changes a little bit. And here you have one where it's actually more complex here, and it looks like it's actually sort of winnowed down and decreased. So this is just an example of how plastic and changing and malleable dendrites are, even within the span of basically about 3 months. This really does suggest that the more you use information, the more you're going to maintain those connections. But if you don't use it, some of those connections will sort of fall to disarray. Here are some examples of the effects of experience on dendrites. So here, before a particular experience, we have here a single synapse right here on a dendritic spine. So we have one right here. Here's axon 1, axon 2, and axon 3. Okay, so here's before an experience. Here's an example of how some of these connections can change after experience. So one, you can have a new axon that comes in. So you can actually have the formation of new synapses at these axon terminals. You still have your original axon. You can have another new axon here. So you can have more synaptic connections. Axon 2 can also have this sort of axon collateral, okay, which basically means that when this neuron fires, then you're going to have double bang for your buck in terms of EPSPs. There's going to be 2 EPSPs coming in here, increasing this, or 2 IPSPs coming in here, decreasing the chances of this firing. And then here you have axon 3, which has remained the same. So this is a really good example of how the structure between neurons can change after experience. Here are some other various shapes of dendrites and dendritic spines that can occur. Branches of 3, they can move out here into different type. You can have a spine sort of bridge on top of another spine. You have 2. Again, here it would be more bridging out. Here's the different types of examples. So there's all different types of ways that your dendrites can change as a function of your experiences.